Okay, so as I mentioned, my name's George. I'm a postdoctoral researcher at University College London. And we're going to talk to you today about how we use a new way of analysing data, which is called machine learning in our UCL research. So specifically, our group work in autoimmune diseases, and that comes under the umbrella of rheumatology. So we work in the Department of Rheumatology. And we're also teamed up with adolescent rheumatology. So these are two kind of separate things, where adolescent rheumatology is an important focus because there's different disease phenotypes between adult and juvenile onset diseases. So let's get started. So if you can see on your screens, there's some instructions to join Sli.do. So all you need to do with this, if you've got a smartphone ideally, but you can also use a separate tab on your computer, what you can do is if you just type in Sli.do on your web browser, just Sli.do, and what that will do is bring up a page where you can type in a hashtag, and here you need to type in the word machine, and that should take you through to our event. Alternatively, what you can do is you can scan the QR code on your screen, and that will take you also through to the event. If you have any technical issues throughout, just get in contact with us. Sorry, I just keep switching between. You can get in contact with us through UCL Rheumatology Engage at gmail.com. So we're going to have, the agenda today will be, we're going to have an interactive Sli.do icebreaker just to get used to using the software. And then we're going to have an introduction from Professor Liz Jury. He's going to tell you a bit about our group and some of the research that we're doing. And then after that, we're going to have a talk from Junjie Pang with a Q&A. And we're also going to have a talk from Leda, who's in our group, with a Q&A. And then we'll have an interactive session where you can get involved through Sli.do, and then some feedback. So I'm hoping that everyone was okay getting into Sli.do. I'll just repeat if people have joined late. What you need to do is either ideally your smartphone, but also if you have a separate tab in your web browser, just search Sli.do, and that should take you through to the website where you can type in hashtag machine, and that should take you through to our event. So what you'll see is a Q&A tab and a polls tab. So throughout the event, you can send in questions to us. Doesn't matter what, there's no such thing as a bad question. Just send in questions, and we'll be able to moderate those and answer them during the Q&A sessions. And we'll also do polls, which will be released throughout, and you'll be able to interact with us through those. I think that's all for now. So I think we can get started. So we're going to start with an icebreaker. So I'm just going to switch us over to Sli.do. On your phones, you should see the Sli.do coming up. So what we're going to do is release a question just to practice run, really, just to try out some questions. So if we can release that question. Okay, so just a silly one to start. Which pet is best, cats, dogs, or neither? So what you'll see, hopefully on your screens, you'll be able to send in your response. Yeah, looks like it's working. I can see the numbers climbing. Just give that a bit longer to see if anyone's responses are coming in. Okay, so we'll release those now just to show you how this works. So it looks like we've got a lot more cat fans in the audience. So we've got 48% of you saying that your preference to cats, a couple of dog fans, and some fans of neither. So that's basically how it works. We're going to have some different types of questions throughout. Some of them you need to type in words, and some of them are just simple multiple choice, like the one you've just had. So we'll start with some more machine learning based questions. So if we can release the next question. Okay, so on a scale of 1 to 10, how would you rate your understanding of machine learning and its applications? You should see on your phones this poll now, or on if you're on a laptop, you can see it on that tab, and just send in your rating. Give that about 10 more seconds so you can get your responses in. Okay, we'll release those then. Okay, so we've got not the best understanding of machine learning, which is great because hopefully we can go through some of the applications of that today. So it looks like the majority of you have gone about 50-50, with quite a few having little understanding, and a couple of people that have got a bit more knowledge about that. So that's good. We've got a bit of a mixed responses there. So I've just got another question to ask you for the preliminary introductory questions. So this one, you just need to type in rather than the multiple choice that we've just had. In as many words as you feel appropriate, what does machine learning mean to you? You can say anything as long as you want. I'll give this one a bit longer just so you can get your questions in. Thanks for sending these in. Really appreciate this. We want this to be an interactive event, so as many responses as we get is great. Okay, so I think we'll release those now. Feel free to send them in. We can still display them while we're showing the results. Okay, so we've got a bit of a mixed bag there. We've got some people thinking it's for big data, which we'll go through today. We've got maths, algorithms, networks, quick processing. See some people hoping that it will help with disease. Got some with nothing at the moment, which is absolutely fine. This is just building up a cloud of words. Essentially, the bigger the word, the more it's been mentioned by you as you send them in. Yeah, keep sending in your responses. The more the merrier. Okay, great. So we'll move on to the next question. So again, this is another one where you just need to just go for the multiple choice. So you can click on as many as you want. So which of the following do you think machine learning can be applied to? Again, if you're joining late, all you need to do is just go into your web browser on your phone or on a separate tab on your laptop or computer and just type in Sli.do, and that will take you through to the Sli.do website. And you just need to type in the hashtag machine, and that will take you through to our event where you can interact with us. Or alternatively, scan the QR code with your phone camera, and that will take you through to the website. We'll give you a link. Okay, so I think we'll release those answers now. Okay, so it looks like the majority of you have ticked all of the boxes, which is the correct answer. Machine learning can be applied to all of these. So we've got research into diseases, which is the way that we use that. Medical diagnosis, behavioral patterns, targeted marketing, and even video surveillance. So it's got a wide range of applications. And like I said, we use it for research into diseases, which we'll tell you a bit about today. So we just got one last question in this icebreaker session. So this is another free text. So if you just send in, why do you think machine learning is so popular these days? So currently, why is it so popular? Again, there's no such thing as a bad answer, so just send in whatever you think. Okay, so I think we'll start releasing some of the answers. Feel free to send them in whilst we're going through some of the things that you said. But cheaper than traditional lab work, helps with learning, new technology, powerful, available to a lot of the use of the word understanding, complex systems. Again, we've got powerful. We can concentrate more on human skills. Thanks for sending in your answers. These are great. Automated data analysis. Too much data. Reduce cost. These are all great points. Advances in hardware. Powerful and cool. Another have no idea, which is absolutely fine. Useful and informative. It sounds cool. Great, thank you for sending in your answers. Okay, so that's it for the icebreaker session. We're going to take us back to the presentation mode. So now we're going to have a talk from an introductory talk from Professor Liz Jury. And then Liz is going to introduce you to Junjie and Leda. And I just want to stress again, there's a Q&A session on your Sli.do. So if you're on your phone or on your laptop, just feel free to send in questions to us whenever you want. And we've got a team of people in the background that will be looking at your questions, and we can release those for the Q&A after each of the talks. So I'll hand you over to Professor Liz Jury now.
Oops. Sorry, I was just unmuting my video. Okay, good morning, everyone, and thank you very much for logging in today, which is our first virtual event or interactive event. Normally, we try to meet with different groups of patients at least once a year, but obviously it's not been possible for us to do this this year. So I lead a team of a group of researchers investigating immune responses into people affected by a set of diseases known as autoimmune diseases. And this includes conditions such as rheumatoid arthritis, lupus, multiple sclerosis, and Sjogren's syndrome. And these diseases are linked by the fact that they have different defects in their immune responses. Now, normally the immune system helps us to all fight things like infections and cancer. But in autoimmune diseases, the immune system becomes active against parts of our own bodies, and this leads to disease. Most of the treatments for people affected by autoimmunity are focused on dampening down the immune responses. And so it's very important for us to understand exactly what is going wrong in each disease, so that we can better target the defective parts of the immune response and find better treatments. One of the big problems faced by doctors is that symptoms and disease features experienced by people with autoimmune diseases can have a huge variation, even within the same type of disease. And also, some people will respond to some treatments, but not to others. So part of our work is focused on trying to group people or stratify them in order to identify who will respond to which treatment. And that will enable doctors in the clinic to treat patients more quickly with the correct treatment and much earlier in the disease course. So to do this, we're gathering lots of different information. This includes really detailed information from patients themselves, about their history, their treatment, all their different types of blood tests, what they eat, as well, and combining this with information we get from the blood, so what's going on with the genes, the different proteins that are circulating in the blood, the different fats, as well as a very detailed understanding of all the different types of immune cells that we have in our blood that make up the immune response. So we take all this information or data and we put it together to try to identify groups or patterns. And this is where the machine learning comes in. So all this research involves a very big group of people, not least the patients themselves, of course, attending University College London hospitals, who put up with this, taking a lot of blood and asking lots and lots of questions all the time. And actually, this research is very expensive because we're collecting so much different types of data. So we're really grateful for the funding of the charities that support our research. But the main contribution on our side is the different scientists, the clinical team, and the administrative staff that really are essential for us to perform all this work. In terms of the clinical group, we have doctors and nurses that provide us with a detailed understanding of the diseases and the different aspects of disease and treatment. The administrative, technical, clinical, administrative staff help to oil the wheels of our research, and we couldn't do it effectively without them, as well as the scientists. And we have different types of scientists, those that you would traditionally associate with research, that go into a laboratory and wear a white coat and perform experiments. But more recently, we've got a group of scientists that work at the computer to do their experiments. And this is what we're going to tell you a little bit more about today. We have two young PhD students who've been doing some research into multiple sclerosis. So that's our first talk from Leda. And the second talk will be from Junjie, who's done some machine learning applied to immune cells from patients with lupus. So I'm going to hand over to our first talk, which is going to be from Leda. Thank you very much.
Hello, my name is Leda, and I'm going to give you first a little introduction to machine learning, and then I'll talk a bit about MS. Oh, doesn't seem to want to move. So machine learning is a method of data analysis which builds mathematical models using large data sets and involving minimal human intervention. So there are two main types of machine learning, and these are supervised, which uses labeled data, and unsupervised, which uses unlabeled data, which I'll go into in a bit more detail. So what you'll often hear thrown about with the term machine learning is big data, And big data is essentially just a large collection of data. So supervised machine learning generally is split into a training group and a testing group. And so in this training group, you provide the data with labels. And so what these labels are, for example, if we look at these groups of shapes, you provide the labels that these are circles, these are triangles, and these are squares. And so what the machine will then do is try to learn what makes each of these shapes different from each other. So it will learn what makes the circles unique from triangles and what makes triangles different from squares, and so on. Once it's learned that, you can then provide it with a testing group. This is essentially just another group of shapes where you don't tell the machine which are which, and the machine will try to use what it's previously learned to predict which shape is which. And it's through this testing group that you are able to actually see how well it's performed, because while the machine doesn't know which shapes are which, you do. So how we usually evaluate how a model has performed is using what's called a confusion matrix. And so in this example, you can see that we know that there are 12 shapes. We know that 4 of them are triangles, 4 of them are circles, and 4 of them are squares. And we want to see what the machine has predicted. So in this example, it's predicted everything correctly. So 4 triangles predicted, of which all 4 were triangles, and so on. But let's say it made a mistake, and it's predicted this circle to be a triangle and this triangle to be a square. This is where the confusion matrix can be helpful, because we can see that, yes, it's predicted 4 triangles, 3 of which were correct, but 1 of them was a circle. We can see it's only predicted 3 circles, but yes, they were all correct. And we can see it's actually predicted 5 squares, 4 of which were correct, but 1 of them was actually a triangle. And so from this, we can say 10 out of 12 shapes were predicted correctly. So the model has an accuracy of 83%. In unsupervised machine learning, you don't give the machine any prior knowledge at all. You just give it the data. The machine will then try to learn what each point are based simply on the data, and try to separate them into different groups based solely on this unlabeled data. This can be a bit more error prone, especially as it doesn't know how many labels there are to begin with. So for example, it might decide that these here are actually ovals, and these 2 squares here are actually diamonds. So for this reason, due to it's more likely to produce errors, and also just it's a bit more complex, we mainly focus on supervised machine learning. So one of the most common examples, or commonly used examples of machine learning, is with irises. So these are all irises, and to me at least, they look very similar, but they're actually 3 different species. So there is publicly available data on these irises, where they look at different measurements of the irises. And each column here is what's known as a feature. So in this publicly available data set, we are looking at the sepal length, the sepal width, the petal length, and the petal width. And so using this, the idea is you can combine or use all these 4 measurements to create a model which can predict what a species might be of an unknown iris. And so the machine learning models that I'm going to talk through today are k-nearest neighbors, decision tree, random forest, and logistic regression. So the idea behind k-nearest neighbors is that you decide, or the machine will decide, what a new iris is based on how similar it is to the other irises it's previously seen. So if we were to plot, for example, petal width against petal length, and we plot all the irises we have in here, you can see that they kind of do separate. So we have the blue here is 1 species, the red here is 1 species, and the green here is 1 species. And the idea is, if you were to introduce 3 new points, you would just look where the points are to decide what that new iris is. So for example, this point right here, is if it's a new iris that you plotted, you can see that it's next to all of these blues, so it's probably an iris setosa. Whereas this point here, you can see it's surrounded by a lot of reds, so it's probably an iris versicolor. This point here might be a bit more ambiguous because it's close to some reds and some greens, but most of the ones that are close to are greens, so it's probably iris virginica. And this is essentially how the K nearest neighbor works. And when we apply this to our iris data set, we get this confusion matrix that I've mentioned before, where you can see that it's predicted only 2 of them incorrectly. Decision tree is probably the most commonly recognized form of machine learning, and the general idea of decision trees is to reduce the disorder within a group. So if you start off with 3 species in this circle, or 3 colors in this circle, the idea is you want to find a split that will separate out the colors so that you end up having just 3 different circles with 3 different colors. And it will make a decision, the machine will make a decision at each point, what is the best cutoff to use to separate out the most. And so when we apply this on the iris data, you can see something that looks like this, where it's trying to find a cutoff at each point to separate out a different species of iris. And when we use this on our iris data set, we can see that this similarly gets only 2 wrong, but as you can see from the confusion matrix, it gets them wrong in a different way. So it still predicts 15 of each species, but it gets 1 wrong in 2 of them. So in a random forest, if we think of the decision tree that I just talked about as 1 tree, then a random forest is simply just a lot of these trees over and over. I won't go into too much detail about this, as Junjie will talk more about this in a bit. But the idea is if you just look at all these trees, different trees over and over, then you can get a consensus of what to do or what decision to make. And the amount of trees can vary, can be 10,000 or can even be 1,000,000 trees to look at. And when we apply the random forest to the irises, we get a very similar result of 2 wrong. A logistic regression tries to find the relationship between the different features and the classification or the different species. But instead of just looking at 1 feature, it will look at all of them. And based on this, we'll try and assign a weight to each feature. And when we apply this model to our iris data set, it performs the best with only 1 wrong. But what's the point of all this? So machine learning can identify important features for us. For example, in what I've just talked you through, we can identify some important features, including the petal length and the petal width. But in addition to this, we can build models where with just a few measurements, we can predict the species of a new unknown iris with around 95% accuracy. But this is just in flowers, so how is this relevant to us? So that's what my project is focusing on. And most recently, we focused on using metabolomics to predict the development of anti-drug antibodies in multiple sclerosis, which I'll explain all. So multiple sclerosis, or MS, is a chronic neurological disease that's driven by inflammatory and neurodegenerative processes. It's pretty common, with 1 in 600 people in the UK affected, and the main treatment options include disease-modifying therapies. Disease-modifying therapies can then elicit an immune response against this treatment, which leads to the production of anti-drug antibodies. These anti-drug antibodies can then alter the response the patient has to the treatment, even rendering it ineffective. So it's important to be able to know as soon as possible whether you will respond to the treatment or not. Metabolomics is the large-scale study of small molecules known as metabolites, and metabolites are the products of metabolism. So the metabolites that we look at are different kinds of fats or different kinds of cholesterol and things like that. And so the aim of my project was to predict the response to treatment in MS patients using metabolomic data. So if we go back to what we did with the irises, I just want to stress that it's the same methods but just different data. So while in irises, we're trying to predict the different species of irises, in MS, we're trying to predict the response to treatment. While in irises, we looked at the plant measurements, in MS, we're looking at the metabolomics. But the methods themselves are identical. We use the same models to try and predict this. And so when we apply this to our MS data set, I was able to develop models to predict the response to treatment with accuracies ranging from 84 to 88%. And this is an example of the decision tree that we've built, which has an accuracy of 84%. But above, also, we were able to identify important metabolites that might influence treatment response, or predict treatment response, such as HDL, LDL, and different proteins. So with that, I wanted to thank you all for your attention, and I'm happy to answer any questions.
Great, thanks, Leda. We're just going to swap over to Q&A now. So on your Sli.do, feel free to send in any questions. Looks like we've had a few come in already. The first one, is there a downside to machine learning?
Yes, so as wonderful as machine learning is, nothing's perfect. So machine learning can be, will rely a lot on interpretation, because the machine itself won't consider all these different contexts or biological contexts that might be involved with all the data. So there will be a lot of human interpretation involved, and with this can come some biases. It also can be hard to get the correct number of people involved with it, because it could need a lot of a lot of patients, a lot of data. But also it might be with some, it's often to have more healthy controls, for example, than someone with MS. So it can be a bit of an imbalance in the data to begin with, which can lead to further bias. So it's just important to bear all these in mind.
Okay, great, thank you. It looks like there's a couple of you that have liked this one, that will slides be made available? Yes, we're hoping to put this recording onto a new YouTube channel, which I'll point you towards at the end. So yes, we'll be able to look at these slides afterwards and rewatch the event. So the next question on the list, what is your favorite thing about machine learning?
I quite like that it can sometimes never be what you think is influencing a model. So like when I was building these iris data sets, or these iris models, I was trying to look at the irises and figure out what might be the feature that predicts it the best, and I was completely wrong. So I quite like that.
Okay, thank you. And the next one, what does machine learning look like? Or what does the machine look like that does the learning, sorry?
So the machine itself that does it, well, it's all done on a computer. And so we usually have these softwares that will run scripts for us. So we will write the scripts and essentially just push enter once we've written it, and hope no errors come up.
Great. We've got, aside from metabolomics, is there any potential to include medical imaging, e.g. MRI, CT, in relation to organ damage in this context?
Yep, absolutely. There are definitely a lot of different studies which do look at imaging data or machine learning in images. There are also ways that you can try and turn images instead to measurements. So we can use, for example, if we want to look at organ damage, we could look at size of plaques and things like that, and that would be measured using an MRI. But there are definitely also methods to use machine learning on the images themselves.
Thank you. Thanks for sending in your questions. These are all great. So how powerful a computer is needed for machine learning, and how long does it take to produce a model?
So how long it takes to produce will also depend on how powerful the computer is. But it depends as well on the size of your data set. So if you're looking at 100 participants, then it's perfectly reasonable to do it on your ordinary laptop or computer. It doesn't need to be that powerful. But as soon as you're approaching larger numbers, for example, genetic association studies will often look at thousands and thousands of people. That is when laptops won't cut it, and it will need to have a much more powerful computer. For how long it takes, again, will depend on the data you're using. So for example, the random forests model I talked about, if you're only using 10,000 trees, for example, it can take a matter of seconds to run. Whereas we've seen people using 1 million trees or more and that needing to run overnight. And definitely with those big data sets that I was talking about before, with the thousands of participants, that can be an overnight job as well.
Great. When do you think machine learning models will be incorporated into routine disease diagnosis and treatment guidelines for major diseases?
Oh, I don't know that. I don't think it's that far off, but I think it will still have someone looking at it. I don't think it will necessarily be automated.
Yeah, I think it needs to take a certain level of trust, doesn't it, between patient, doctor, and machine. Okay, great. And then we've got a few questions left. In terms of medical data sets, what would be an acceptable error rate?
So I think usually if you have an accuracy around 75, above 75, it's all right. Above 85, it's getting better. Ideally, of course, you want something in the 90s, but it's also just acknowledging the size of the data sets, the rarity of the disease you're looking at, and things like that. So it sort of depends on the different context, but definitely not for use in clinic would anything under 90% be used yet. But certainly for research, it can be a bit more flexible.
Great, thank you. And then the last one, do you expect machine learning to become even more accurate in the near future?
I think the more we understand about the context of disease and therefore to make our interpretations, it will get better. The more we can include more people into machine learning, it will get more accurate. So yes, I think it will get even more accurate as we go on.
Great, thank you so much, Leda. And thank you for sending in your questions. We're going to move on to our next speaker now, Junjie, and please feel free to send in questions during his talk. So I'll just sort out passing over to Junjie and then I can hand over.
Thank you, Leda, for the great talk. So hi, everyone, I'm Junjie, and I'm glad to be here today. I'm a junior researcher and a PhD student at UCL, and my current study focuses on using machine learning and statistical approach to understand autoimmune disease. And today I would like to use one of our study of juvenile onset lupus as an example to illustrate how machine learning can help with our research. And the title would be Machine Learning for Precision Stratification of Juvenile Onset Lupus. So first, we have some background about the disease. Juvenile onset lupus, or JSLE, is a rare autoimmune disease and is onset before age 18. It's a female-dominant disease with a ratio of 4.5 to 1. And juvenile onset lupus is only account for around 15 to 20% of all lupus cases. And currently, there is no cure as well as age-specific treatment for the juvenile lupus patients. And studies have shown that compared to the adult onset lupus, juvenile onset lupus has a more severe disease presentations And because JSLE is a disease with high immunological heterogeneity, which means the immune cell construction is highly diverse within the JSLE patients. And because of this variety in patients, it eventually leads to poor treatment outcomes for specific groups of JSLE patients. So in order to tackle this unmet clinical need of JSLE, it is important for us to find a way to stratify patients based on their own immunological profile, and therefore able to provide a precise treatment options for each individual. So in our study, in order to investigate the immune profile of the JSLE, we need to first collect data of immune cells from both lupus patients and the healthy controls. Blood samples from 67 JSLE and 39 healthy controls were collected from UCL Hospital. And because this is such a rare disease, being able to recruit this amount of patients is actually crucial for the study. So after collecting the blood samples, we use a technique called flow cytometry, which is able to identify and quantify different cell types in the blood. And in our study, we measure a total of 28 immune cell types that covers the most of the important cell types for autoimmune disease. And after collecting the immune cell profile, how do we know which specific immune cell types are actually important for the disease development? And so this is where we introduce the machine learning approach, often known as the powerful black box that perform magical things that humans are not capable of doing. And by applying the machine learning approach, we can then select the most critical immune cell subsets. And then by applying these immune cells as potential biomarker, we are able to stratify the patients into different groups for a more personalized treatment. And so the major machine learning algorithm that we use in this study is called random forest. And Leda has already briefly introduced random forest in her sessions. So random forest is a supervised learning methods for classifications. And just like a forest that is made up of a large number of trees, random forest is also made up of a large number of decision trees. And for each of our decision tree in our random forest model, it is constructed by a random subset of our original data, which will be the immune profile of the JSLE and the healthy control samples. And in this model, we want to use these immune cells informations to predict if the samples are from juvenile lupus or healthy control. And so for each splitting point of the decision tree, conditions will be randomly selected to decide which way it goes. And in our case, this would be the immune cell selected from our 28 immune cell types. And at the end of each decision tree, the classification result will be generated as either identifying the samples as lupus or healthy. And this process will be repeated until our forest is big enough to generate a stable result. Here in our study, we used a total number of 10,000 decision trees to build the random forest model. And finally, by considering all the results from each decision tree, we are able to get the final classification result, which will be predicting each samples as lupus or healthy. And after building our random forest model, we can then validate our model performance using the validation data set. And the result shows that our random forest model is able to achieve an overall prediction accuracy around 91% for separating lupus samples from healthy control. And more importantly, our random forest model is able to rank the importance of the immune cell types so that we are able to select the most contributing immune cells for the model, therefore selecting immune cells that are most critical for the development of the juvenile lupus. And also to make sure the selected immune cells are reliable, we also validate our results by performing other machine learning models and statistical tests. But due to the time limit today, I'm not able to go through all of them. So eventually, if we're combining all this analysis, 8 out of 28 immune cells were selected as potential biomarkers. And by using the selected immune cell, we then stratified the JSLE patients into 4 groups with distinct immune profile and potentially provide more effective treatment for each group. And finally, by analyzing the longitudinal clinical data, we are able to predict the long-term outcome of the lupus groups. Specifically, we found out that the patient group 1, which is showing red here, they have the highest average disease activity scores. And if you look at the 5-year follow-up study on the right-hand side, patient group 1 also has consistently having a more active disease. And so this result suggests that despite most of the patients having the same treatment routine, patient group 1 still has a more active disease, which is likely to be associated with their distinct immune profile. Therefore, it is essential for us to conduct a more careful disease management for those patients. And in conclusions, our study used machine learning approach to help understand the immune cell profile of juvenile lupus. Our random forest model can identify JSLE from healthy control with a 91% accuracy, where a important immune cell subset was selected. A group of lupus with distinct immune cell signatures are recognized in our study with high disease activity. And also further study is essential for discovering optimal treatment options for those stratified groups. And last but not least, as a person with great passions in machine learning and research, I always believe that the concept like machine learning can be less scary and more enjoyable if we add the touch of imaginations. So allow me to finish the talk in a different way of concluding. And so the study is about the juvenile lupus. And it is surrounded by this artificial but beautiful random forest. And you can see the juvenile lupus is in quite a good mood. This is because it knows with the help of the powerful machine learning technique, we start to understand the disease better. We're able to select crucial immune cells for JSLE development and use them as biomarkers to stratify the patient precisely. Treatment will become more effective and personalized for every juvenile onset lupus patient, and symptoms will become more manageable. And hopefully in the near future, JSLE will no longer be an incurable disease, while machine learning will continue helping researchers to tackle other complex diseases. And that's it.
Great, thank you, Junjie. Great talk. So we've got another Q&A with Junjie now, so please feel free to send in more questions as we go. The first question we've got here is, should I be concerned about data privacy?
So I think data privacy is one of the most popular concerns when people talk about applying big data and machine learning. And I do believe that data privacy is super important, especially when participants are helping the research by donating their samples. And so I think in order to make sure that the data is secure, we have this very strict consent and data protection process where people are trying, when people are trying to participate in this type of study. And also after we collect the samples, I think the personal identity will be replaced by numbers and code. And so that when other people or researcher like me, when I get the data, there is actually no way for me to figure out who is who. And also I think UCL and other research institute, they really value the data privacy a lot. And using machine learning in the study, I don't think it will change anything about that.
Great, thank you. You got some fans. Fascinating talk. Thank you, Junjie and Leda. So the next question is, does your lab work with colleagues in other autoimmune disease areas? For example, juvenile idiopathic arthritis, JIA, through cluster study at Gosh and Manchester. So I can have a go answering that. Yes, we do a lot of work with the Adolescent Centre. So there's going to be a huge amount of data coming from the cluster study, which is between Gosh and Manchester, as you can see here. So yeah, it'd be great to get involved and try and apply these kind of analysis methods as a collaborative effort. So yeah, that's definitely something that we're looking into for the future, and it's a great, the cluster project looks really great. Okay, so the next one, so the decision trees are like algorithms.
Yeah, that's true. Yeah, I think all the model that we have bring up today is kind of like sort of algorithm, and you can also see it as model. So it really depends how you see it, and so deep down is all like mathematical model using different algorithms.
Okay, thank you. Do error margins in machine learning potentially help indicate new disease subtypes, and could it change how we recognize lupus in the future?
Yeah, I do think, yeah, it's quite like a good point. I think in our study also, at the end of our study, we already stratified 4 patients groups, and I think we need further study to have more clinical proof to say that if they are a disease subtype. But definitely, it could be quite useful in disease such as lupus, because it's so the heterogeneities of lupus is very high, and it's a quite complex disease, and so really patients with different symptom and disease presentations, as well as their immunological profiles, if we can separate them into different subtypes, that would be super useful for clinical.
Great, thank you. The next one, is there anyone developing new drugs to work alongside stratification, or is the focus on existing drugs in a more targeted way?
I think it could be both, and obviously, if we can stratify patients in a way that each group of the patients can be benefit from the existing drugs, that could be very effective and time-consuming and money-consuming. But then, if we eventually discover a new pathogenesis in specific group of patients, then researchers might get inspiration from it and eventually develop for new job for them.
Okay, great. Do you expect to gain a completely different result from machine learning when the sample size expands from 10 to 100?
I think the answer is no. Technically, it should be no. Because if you, let's say, if we are using the random forest model in our study, the first thing we need to make sure is that our result is stable. So it's not changing every time we run the analysis. So by doing that, in the random forest model, we can increase the decision tree numbers to a very large size, and eventually the result will be the same as no matter how many times you run it. And also, but in terms of the sample size, if it increased a lot, definitely machine learning prefer a bigger data, bigger sample size, because it ensure the robustness of the model. And it also depends on the data quality of the data that we collected. So if the larger data has a low quality of the data, and the results will be not as good even as the small data set.
Okay, thanks. Let's try and get through some more questions. Do you think the widespread use, are you looking at other diseases? Switch.
Wait.
Let's see which one I go for. Okay, so are you looking at other diseases in addition to see if there are commonalities across disease entities leading to particular presentations?
Yeah, actually, we are doing one of our study is about Sjogren syndrome, which is a completely different autoimmune disease. However, we do find a lot of overlapping immunological features between the Sjogren syndromes and the juveniles and the lupus patients. So we are working on that, and we try to stratify the patients despite of their diagnosis, and hopefully we can find like the commonality between them, and to see if we can share some of the treatment between the groups.
Great, thank you. So the next question we've got, do you think the widespread use of electronic health records can help rapidly boost the potential of machine learning?
Yeah, I think definitely. As I said, machine learning really depends on having a good quality big data. So being able to apply the electronic health records and use that, use machine learning to analyze those data could be just really powerful to analyze a lot of disease, analyze a lot of clinical problem. And I think that is also a promising future for machine learning in clinical research as well.
Great, we'll just get through these last three questions and then we can move on to the next part. What do you take into account to choose a machine learning model over another? For example, random forest versus logistic regression, or for example, other models?
Yeah, so basically each machine learning model have their own advantage and disadvantage. And I think the principle is that in most of our study, we usually applied more than one approach, just to avoid the bias for using only one specific machine learning approach. And also the performance of the machine learning model could be quite different even when we are using the same data. So it is actually quite necessary to try out a lot of different methods and see if they performed well in these like specific studies.
Thank you. Do you know what the current role of machine learning software in the software is used in personalized medicine to issue recommendations for the best therapeutic choice?
Not so sure about this, to be honest.
I think it's going to be an upcoming thing in the near future that they'll start using these kind of techniques in personalized medicine. But at the moment, I think there's still a certain level of maybe anxiety about the fact that you're putting your trust into a machine for a medical diagnosis. But I think as these models become more accurate and more published, I think you'll see more of that in personalized healthcare. And then the last question, how would you differentiate between precision stratification in your title and accurate stratification?
Yeah, so I think the idea of precision stratification is all about identifying the underlying common features of the disease and divided the populations into groups based on those features. So instead of just using one treatment routine for all the patients, each divided groups should be benefit from a precise treatment options. And so with that being said, whether the stratification is precise enough or accurate enough really depends on whether each patient eventually receive the right treatment options within their groups.
Perfect. Thank you, Junjie. That's a great talk, and thank you for
George.
answering the questions, and thank you for sending them in. We're going to go on to the last part of the session. Sorry, Junjie. So this is we're just having one more interactive session. We just got 4 questions. So if you could just get involved for this last part of our PPIE event, that'd be fantastic. So we're just going to release some questions following our talks. So if you can pick up your phones, hopefully you can see the polls coming up. If you've lost track of Sli.do, then just type in Sli.do into your web browser, and you can join using #machine. Or alternatively, you can just scan the QR code in the corner of your screens now, and that with your phone camera, and that will give you a link to answer these questions. So first of all, do you think machine learning overall is a good thing? Yeah, just give you about 10 more seconds to get your answers in for that one. Okay, so let's see what you've put for that. Oh, fantastic. It looks like perhaps we've convinced you in some of our research that it's going in the right direction and that it's beneficial for both our research and the future of healthcare. Okay, so the next question. So this is just a scale for your rating. So following this event, on a scale of 1 to 10, how would you rate your understanding of machine learning and its applications? Okay, so let's see what you've put for that one. Okay, so it looks like the understanding's gone up since the start of the event, so that's great to see. Like we're doing our job in some form. So we've got an average of, well, the highest rating was 41% of you, but there's a few of you that have gained a much better understanding of machine learning from this, which is great to see. Thank you for sending in those. Our next question is, regarding machine learning in research, do you have any concerns about the following? So you can tick multiple if you feel that you have more concerns. Okay, so let's see what you put for that one. Okay, so the most popular response was bias in data, also quality of the data being analyzed, data privacy, and then you've got quite a few of you still saying data processed by machines instead of human beings. And so there's actually quite a small number that have no concerns at all about machine learning. So that's possibly why there is a lot of debate about whether it should be used in personalized medicine and things like that, because there's still a lot of uncertainty about whether data's being put into the machines. Maybe it gets lost in translation. There's a lot of bias in the data, perhaps. So I think it just needs to be built a level of trust and get some more solid data that this is a good way to go for personalized medicine. Okay, so just our last question for you today. So this is our first public engagement event. So this is the first in a series that we're hoping to do over the next year or so. So these are some examples of events that we could follow this up with. So if you could just vote on ones that you would like to see us do. So the examples are from clinic to the lab, what happens to blood samples in autoimmune disease, microscopes, fluorescence, DNA and beyond, how do we look at immune cells, what are autoimmune diseases and how do they overlap with each other in research. We can do a meet the team session, showing you how do different occupations come together in research. How different is the immune system in males compared to females, which is something that we work on in our lab. Does age matter in autoimmune disease? Why do we need adolescent-specific research? And then finally, personalized medicine, how research is changing the one-size-fits-all approach to healthcare. You can tick more than one if you'd like to see more than one of these in the future. But it's important to us that we target these events towards what our audience want to see. Just give that a bit longer for you to get your answers in. Okay, so let's see what's come out as most popular from these. Okay, so a lot of you interested in personalized medicine and how research is changing the one-size-fits-all approach to healthcare. So that's been the most popular response. Does age matter in autoimmune research? That's great because we are heavily, a lot of our group focus entirely on adolescent-specific research. So we'd be happy to show you some of our research regarding that. Next one down was what are autoimmune diseases and how do they overlap with each other in research? And we've got a pretty similar response engagement to look at from clinic to the lab, what happens to blood samples, how different is the immune system in males compared to females, and then meet the research team with 44%. So it looks like all of the topics are of interest. We've got a few that are much more popular than others. So thank you very much for sending those in. So with that, I'd like to say a massive thank you to everyone who joined us today. If you want to find out more about the kind of things that we do in our lab and follow us, you can follow us on Twitter. That's @jury_lab or @UCL_Rheum_AYA. That's our Adolescent Centre Rheumatology account. So we put up lots of tweets showing what we're up to in the lab and different publications that come out from us. We've also got a new YouTube channel which we're hoping to put these events on. And so that's UCL Rheumatology Engage. On your Sli.do on your phones, in the top left corner, there's the 3 lines to bring up the menu, and you can actually find links to both the YouTube account and the Twitter account. So you can just directly link yourselves up from there. And please let us know on this same Sli.do event, we're going to release feedback to your phones now. It would be fantastic if you could just provide us with some feedback from our event today. That we'd really appreciate that so we can improve future events, and hopefully everyone enjoyed it. So just keep an eye on your phones and you'll see the feedback questionnaire be released and you can start to fill that out. So with that, I'd like to say thank you, and we'll hopefully see you all soon.